this is going to be a video following up on the in-class assignment we did last week, drawing the steps in the differentiation of muscles. And at the end of that, we did a couple of a couple other drawings, and one of them was this one. I saved a screenshot of it, of me trying to show how we can have a transcription factor that gets expressed. The protein is there, but then there's a delay in the activation of that protein. And our focus is going to be on proteins that are transcription factors. There are lots of proteins that are present and they're delayed in their activation. For example, we learned about caspases. Those are proteins that are in our cells right now and that cause apoptosis, programmed cell death, but they're not active. So there's a delay in their activation. In our case, in our course, we're going to focus a lot on this class of proteins called transcription factors. Many of them have a delay in their activation that is central to development. So we inherited from our parents all the DNA we need, all the genes we need. We're going to turn them on over time and location and amount. But what causes that to be played out over time with delays and orchestrations and timing? And the answer is lots of times it's the regulation of these transcription factors. So I emphasize that muscle differentiation is a great way to start the course, because when the MyoD transcription factor gets made, it just goes back into the nucleus, binds to the enhancer of myogenin, and it activates its expression. It's not delayed at all, really. It's just doing its job. Same with myogenin. It just does its job. So estrogen hormone is a good example because we all have some familiarity with it. What I need to do, I think, based on questions after class last week, is just give you a little background explanation for the estrogen story. So estrogen is a hormone, and it's made in the ovaries during puberty. And it's made in some specific cells called granulosa cells, and those are the cells that make it. Then the hormone is released in the blood, just floats around in the blood, and it targets certain cells. And there are lots of target cells because in puberty our bodies change a lot. And I should say again that I'm focusing on estrogen. Estrogen is the female sex differentiation hormone in all vertebrate species. And I'm not talking really about testosterone, which is the male sex differentiation hormone in all vertebrates. And we can just talk about one of them because they really have the same mechanism of action. So the hormone's going to float around and it's going to affect target cells. And we're just going to pick on the brain. So during puberty, there are changes in the brain. So now we're going to just go back and encourage you to draw along with this as you study for the exam. We're going to go back to looking at this drawing. And okay, and so we had a drawing before, but now just to help with this drawing, what we're going to do is we're going to say this is a cell and it's in the ovary, and I won't write down that word granulosa cell again. It's just a cell, and this cell is going to synthesize estrogen and release it into the blood. So there's estrogen, and it's a small steroid hormone, small molecule, and this cell is going to synthesize it. So we have a general term for this in developmental biology, and this is called a source cell. And we're not going to talk about source cells very much because we're really interested in what that hormone does to other cells. And this is going to be the other cell, and it's going to be the basis for our big drawing. So this is going to be one of these brain cells that estrogen is going to affect during brain cells that estrogen is going to affect during puberty. And this is the cell membrane. Okay? And there are lots of these cells in the ovary. There are lots of these cells in the brain, and so on and so forth. This cell has just a general term, and it's called the target cell. And it's an estrogen target cell. Estrogen doesn't affect every cell in our body, but it affects many, many cells. and we call them target cells. So very general terms, source cell and target cell. Then developmental biologists call this a signaling factor because it's bringing a signal. So it's a great word, it's a general word. And we're going to talk about lots of signaling factors. Really not going to talk much about estrogen. You'll see there are several others that become the big focus of our course. But we're going to talk about it now because it's a great example, because in the case of estrogen, estrogen's interesting when if you look at its molecular structure, estrogen is both hydrophobic and hydrophilic. And that means that when it arrives at this target cell, it can actually just diffuse right through the cell membrane and into this target cell and just float around in the cytoplasm of the target cell. And it has a receptor. And the receptor for estrogen is inside the nucleus. So there's the nuclear membrane. And the estrogen is going to float right through a nuclear pore right into the nucleus. And there it is in the nucleus. And it has a receptor. And the receptor for estrogen is called the estrogen receptor. There it is. It binds. And that's the estrogen receptor. Okay. So it's abbreviated ER, estrogen receptor. And it gets activated when estrogen binds to it. It's a transcription factor. I mentioned that prior to puberty in young girls, and also throughout the life of male vertebrates, the estrogen receptor actually functions as a repressor of transcription. So it's bound to these enhancers on target genes. And so there are a bunch of target genes. And this is, we are not going to learn the names of any of the target genes. But here's an enhancer, and it is an enhancer that the estrogen receptor is bound to. So in very early in life, the estrogen receptor gets made, and in childhood or in juvenile, non-reproductive juveniles of any vertebrate species, it's repressing these genes. And then when the estrogen arrives, it changes and it's now activating these genes. So this is something developmental biologists like to learn about. It's a molecular switch, like turning the lights off and on. And this one estrogen receptor protein does both. It keeps the lights off and then it turns them on. And this gene, again, we could name them, but there are hundreds of them. And so people just generally refer to this as an estrogen target gene. And we're going to use that word a lot. And it's an accurate term, and we just need to be aware that this is this estrogen target gene. And it's just a gene, like the myogenin gene. It's going to get regulated, and its transcription is going to get regulated. And so when estrogen shows up, this gene is going to start to get transcribed. And let's say it's a coding gene, so it's going to start making mRNA. And that mRNA is going to go out of the nucleus, and we're going to make this protein. And it's going to have something to do with the role of the hormone in stimulating puberty. So a really good thing to know is that in the absence of the estrogen hormone, this mRNA is not getting made, and this protein is not present. So all the way through childhood, the estrogen receptor is present, but it's not activating the genes that are the target genes. It's actually repressing them. What we're most interested in here is the idea that the estrogen receptor needs a molecule to serve as its switch. There's a general term for a molecule that binds to a protein and regulates the protein. And the general term is one we'll also want to learn. And the general term is, it's called a ligand. So a ligand is a small molecule that binds to a protein and affects that protein's activity. Okay? All right, so that's where we're at on that. Now, the last term we're going to use here is, you know well, that estrogen is a hormone. And there's another term we're going to learn for hormone. It's a synonym of hormone, and it's called an endocrine factor. And an endocrine factor is a hormone, the same thing. They float throughout the body, and there are lots of endocrine factors: insulin, epinephrine, which used to be called adrenaline, and so forth. They're all hormones, and they go throughout the body. Okay? So we have a very good specific example that came up when we wanted to talk about how a transcription factor can get made, but not do its job right away. It's waiting, it's ready, it's poised, it's a light switch. It's going to get switched on. All right? All right, so that's our example, and now let's use that example to move forward into some stuff, okay? So here's something, and this is a figure from chapter 4. You don't need to look at it for the quiz, and in fact, we'll never learn about this figure. This shows a hormone that's called retinol, and retinol is it's a hormone that's made from vitamin A, and it's one of the main reasons we need vitamin A in our diet. We also use vitamin A and retinal in our vision as a pigment to detect light. This is a hormone, and it's called retinol. Another name for it is retinoic acid. I'm naming those not because we need to know them, but just because you may have heard of this hormone. So it's a hormone. It's an endocrine factor. And let's just look at this. And again, this is not going to be the centerpiece of our quiz. It is just to show you that there are other hormones that behave like estrogen. So this is a cell, and this would be the source cell, and it's making this hormone, and it's releasing it. And so this is a hormone, it's a signaling factor. And you can see this hormone goes, like estrogen, directly into the target cells and into the nucleus, where it binds to its receptor, which is called the retinoic acid receptor. And that receptor is a transcription factor. So here's another delay, and it's in the same category or class of receptors, where the receptors are inside the cell, and inside the nucleus, the receptors are transcription factors. It shows a nice direct regulation of target cell gene expression by this source cell. Right? So the source cell is going through development, and it's changing its own gene expression, and that includes making this hormone that's going to change the expression of genes in the target cells. All right, so estrogen and retinoic acid fit together into a class of hormones and other molecules that are in our bodies, human bodies, and their receptors are all in a group of receptors that are called nuclear receptors. And nuclear receptors are in the nucleus, so that's a great name. In humans, there are 48 nuclear receptors. There's a nuclear receptor for estrogen, the estrogen receptor. Then there's, of course, the testosterone receptor. Then there's this retinoic acid receptor. There's a thyroid hormone receptor, a vitamin D3 hormone receptor, and several more for a total of 48. And they all have the same feature in that the receptors are already in the nucleus playing a role to turn genes on and off. Okay, like that. So just showing you another image of this that we're learning about something that's a general, but actually not a big part of our course. These nuclear receptors are very important, but hormones are not a big part of our course because we're going to look at earlier stages in development. Okay, so I also wanted to show you this, that when we start talking about these signaling factors, they're actually covered in intro bio. Different instructors cover different topics in intro biology, somewhat unfortunately, so I don't know whether you had this, but at USM we use this textbook, Campbell's Biology, and then that textbook, the topics that we're talking about right here are in chapter 11. And it's about this quiz. You don't need to go find a copy of Campbell. So cell communication. So this cell is communicating with that cell. This cell is a source cell. It's going to release a signaling factor, and that's going to affect the target cell. General terms. And again, people always just draw one cell, but there are thousands or millions of these cells that are playing these roles, okay? So just to show you a little background to this, for example, here's epinephrine. It used to be called adrenaline, and when the deer gets scared or a human gets scared of something, like a cheetah, either one of us, they're like, I'm going to run away. And it gets excited, and it can feel that, and you do too. When you're scared, you feel that rush of adrenaline, rush of epinephrine. Epinephrine is a hormone that goes throughout our blood. It's released from a source cell, source cells, goes through our blood, binds to a receptor, and so here's something we want to introduce that's different. This receptor for epinephrine is not a nuclear receptor. It's not in the nucleus. Every receptor, basically almost every receptor, except the nuclear receptors, are actually in the cell membrane. So the signaling factors that we're going to learn about in our course are in the cell membrane. And it needs to communicate the signal into the cell, and it does that through a pathway, a biochemical pathway within the target cell called signal transduction. So the ligand binds to a receptor, in this case in the cell membrane, and its presence is communicated into the cell by signal transduction. This just shows the cellular response. In this case, the epinephrine is not activating changing gene expression. That's common. The cellular response is to break down glycogen and produce a bunch of glucose inside cells. This target cell could be muscle. And the muscle suddenly gets its own sugar rush from stored sugar that was quickly broken down by this activated enzyme to release that energy. So just an analogy or example from IntroBio. Also from IntroBio, this general drawing for receptors that are in the cell membrane, so not nuclear receptors. We have the signaling factor binding. It's a ligand that binds to this receptor protein that stays outside of the cell and activates a signal transduction pathway that leads to a cell response. In developmental biology, we're really especially interested in changing gene expression because the cells are going to get different and they have all the genes, so they must get different by changing gene expression. So we're really interested in transcription and the regulation of these transcription factors by signaling molecules. Okay, here's, I think, the last slide from IntroBio. So we talked some about this, long-distance signaling. And here's the source cell, and here's the target cell, like that, okay? And this could be estrogen, testosterone, epinephrine, like that, and they're hormones. And so all hormones fit into this. We're not really going to talk about hormones in our course because we're going to look at the earlier stages in development. So instead of that type of signaling factor, instead of one that goes throughout the body, we're going to study a lot of local signaling. And a good example of local signaling is when you get a cut and you have a scab. Under that scab, the skin is healing. It's local healing because the platelets that help clot the blood and form the scab, those platelets also release a signaling factor that binds to a receptor in the skin cells. And the signaling factor is called platelet-derived growth factor. It binds to the platelet-derived growth factor receptor, and that stimulates the skin cells to grow quickly, to go through the cell cycle and heal quickly. So these are not endocrine factors. These are called, and this is the term that we want to really remember, paracrine factors. Paracrine factors are signaling factors that go short distances. Just a few cell diameters away. They only go a short distance outside of the cell, the source cell, because they get broken down. They get broken down really fast. They get degraded, inactivated really fast. And so hormones and paracrine factors are very similar. Variety of molecules can play the role of a hormone or a paracrine factor. They're all ligands. They're going to bind to a receptor, a small molecule binding to a receptor. The difference is that paracrine factors get broken down quickly and stay local. Endocrine factors make it throughout the body. Okay, great. This example from our first chapter is an example of a paracrine factor, a local signaling factor that is called sonic hedgehog. And and that's an odd name, but sonic hedgehog is a very important protein in our bodies, and there are orthologs of sonic hedgehog in most animals. Only our most distant relatives don't have orthologs of sonic hedgehog. And you don't have to memorize this. It's in our first chapter, but you don't have to memorize this. Just notice this, that sonic hedgehog would have be made in a source cell. Sonic hedgehog's a protein. So in this case, there's a sonic hedgehog that must get activated in this source cell. And the source cell must be right here because it's a local paracrine signaling factor, and it's going to float a little ways away and bind to this target cell. So again, we don't talk much about the source cells, but it's good to know that they're there. That's the source of these signals, and then this signaling factor, this ligand, binds to a receptor, and a signal transduction pathway affects transcription in this case. So see, we have this general model is how sonic hedgehog does its job in regulating the zipping together of the face. And we learned last time that it's these neural crest cells that are playing the roles of zipping the face together like that. Okay, so here's a figure from chapter 3, and it's full of information, and we need to pick and choose. So we're going to pick and choose today. One of the signaling factors we just learned a little bit about is sonic hedgehog. So that's what this is about. This row is about sonic hedgehog. There's sonic hedgehog, there's its receptor, and this is what some of the roles are that it does in development. We're going to have the most central signaling factor that we're going to learn about is called Wnt. And Wnt is a member of a gene family in humans. They just call them Wnt 1, 2, 3, 4, 5, 6, 19 total. It has a curious receptor named Frizzle. Lots of these genes were first discovered, orthologs of lots of these genes were first discovered in fruit flies, and fruit fly geneticists give genes silly names. And then lots of roles in development. So we're not going to learn very much about the estrogen receptor and that estrogen signaling pathway. We're going to learn a little bit about sonic hedgehog and that signaling pathway. We're going to use as our favorite example Wnt. And we're going to have a drawing assignment on Wnt next week, because the Wnt drawing assignment will be similar to the muscle differentiation video. We'll refer to it throughout our course many times. And we will learn some about some of these others, but we're going to pick and choose and focus our learning. Okay, I want to just show you this. In our syllabus, I included a paragraph from a student's writing who wrote a term paper many years ago for this course, and they talked about learning about this organism. And this is one of these organisms that's very distantly related from us. So there's an evolutionary tree, and here we have sponges, and here we have jellyfish, and this little organism is a hydra living in the ocean, just tiny little inch-long hydra, and so very distant relative as ours, and it has a Wnt ortholog. And we'll refer to this as the Wnt signaling pathway. Okay? And the Wnt signaling pathway, Wnt is a protein, and there are orthologs of Wnt in this organism and every other animal on our planet. And not just Wnt, but this Wnt signaling pathway. So let's take a look at this. This is from chapter 3, and we're going to have a drawing assignment where we draw and learn about this. So this is just drawing the target cell, and it's drawing the target cell when Wnt is absent and Wnt is present. And we're not going to learn all about this, but just for now, and we don't need to learn all about this for the quiz, because we're going to have a drawing assignment. But just for now, I'm going to show you, yep, there's a receptor, and there are proteins in the signal transduction pathway, and then there is a transcription factor, and another transcription factor. And those are what we'll learn in a future assignment. But you should be able to draw a general drawing of a target cell releasing a signaling factor, binding to a receptor on the cell surface, and then a signal transduction pathway to regulate gene transcription. And we don't need to label those with specific protein names, but we should be able to draw that, a drawing assignment like that. Interestingly, just to say this, and it's not going to be part of the quiz, but this transcription factor, you can see what happens is in the absence of Wnt, the transcription factor gets made and just gets destroyed. It gets degraded right away. And the role of Wnt is to stabilize this transcription factor so it can do its job. This is a great example of a transcription factor that has a delay in its activity. So MyoD gets made and does its job. Myogenin gets made and does its job. This transcription factor, beta-catenin, gets made but doesn't get to do its job because it just gets destroyed right away. And it gets destroyed until Wnt shows up and stabilizes it and allows it to do its job. So imagine these dominoes falling, that analogy where one transcription factor activates another transcription factor. Well, this is that, and just imagine the domino gets made, but it is not on the next domino yet because the domino gets made and for a while it just gets destroyed. And you can just watch it. It's there, you know, kind of starting to fall, but it's getting destroyed and can't knock down the next domino until Wnt binds to the receptor in the cell membrane. Notice that Wnt isn't going inside the cell. It's a different kind of signaling factor than estrogen and that retinoic acid. This is the typical kind of signaling factor where the receptor is in the cell membrane and we have to have a signal transduction pathway to communicate the presence of the signal into the cell. Here's just another example. This is from chapter 4, and it's an example of a signaling factor, and our book has lots of drawings like this where it shows the signaling factor in the and it's doing its job like that. And I guess it's showing actually not plus or minus, it's showing a variety of signaling factors that fit into this big class. I'll just show you this big class here. This class, it's showing a figure of that. And we're going to learn some about that class also, but not as much. Here's another example where there's a signaling factor and we're learning about a signaling pathway that again affects the transcription factor. In this case, the transcription factor is inactivated until it adds a bunch of phosphate. You don't have to memorize this or look at it a great deal. I'm just giving you a look ahead in our course and how frequently we want to learn about this. And we'll introduce it. I think that might be it for our video and our quiz. So in our quiz, you should be able to draw that better estrogen drawing that I did with the ovary cell and the brain cell, a source cell and a target cell, showing estrogen being released from the source cell and going right into the target cell and right into the nucleus and binding to the estrogen receptor that then activates the transcription of these target genes. Yep. And then the other drawing you should be able to do is just a general drawing of a source cell and a signaling factor, and in this case binding to a receptor in the cell membrane, and then a signal transduction pathway. You don't have to draw it out in detail or label it, but know the term signal transduction to regulate activity of a transcription factor. So we just want to start getting used to that image and being able to sort of see it in your mind, definitely. Okay, so I'm going to stop here and we'll roll forward. Great, thanks.